Ever pondered how researchers choose outcomes in a drug discovery project? It's a complex web, a labyrinth of intricate decisions and meticulous planning. Every drug discovery project is a journey into the unknown, a quest filled with countless possibilities. Researchers must anticipate a vast array of outcomes, each with its own unique implications. Why do they choose specific outcomes, you ask? Well, these decisions are critical for the overall success of the drug development process. They are driven by factors such as the nature of the disease, the drug's mechanism of action, and the desired therapeutic effect. It's a delicate balancing act, where every choice can tip the scales in favor of success or failure. It's not enough just to find a new drug. It must also be safe, effective, and beneficial to those who need it most. Choosing outcomes in drug discovery is not a random process, but a strategic one, rooted in years of research and understanding. Once chosen, how do researchers measure these outcomes, and who holds the responsibility? In the labyrinth of drug discovery, outcome measurement is a cardinal point. It's a meticulous process requiring precision and accuracy. Various methods are employed, from lab tests and clinical trials to patient questionnaires and beyond. Each method is tailored to the specific outcome being measured, whether it's the drug's effectiveness, its side effects, or its impact on quality of life. Who holds the responsibility for these measurements? Meet the clinical research teams, the unsung heroes of drug discovery. They comprise skilled professionals from lab technicians to biostatisticians, each playing a pivotal role. Their collective expertise ensures accurate and reliable data collection. And then, there's the question of when. Measurements are taken at critical junctures throughout the drug discovery process. Preclinical tests, clinical trials phases, post-market surveys. Each stage requires its unique set of measurements. Accurate measurement of chosen outcomes is a vital cog in the wheel of drug discovery. Is there room for bias in this scientific process? Indeed, bias can creep into the most meticulous research, including drug discovery. This bias could manifest in several ways. For instance, there might be unintentional favoritism towards certain outcomes, or the person measuring the outcomes could inadvertently influence the results. Such biases can skew the results, leading to inaccurate conclusions about the drug's efficacy. But fear not, there are measures to prevent, minimize, or control for these biases. Blinding, where the person measuring the outcomes doesn't know which treatment group the subject belongs to, is one strategy. Another is randomization, where subjects are randomly assigned to treatment or control groups, reducing the likelihood of bias. Moreover, to maintain the integrity of the process, an independent committee could oversee the measurement of outcomes. This way, we ensure that the results are as objective as possible. Despite the potential for bias, rigorous scientific methods ensure the reliability of the drug discovery process. So, how does it all come together? Let's take a step back and look at the big picture. Drug discovery is a complex, multifaceted process, and the outcomes we choose to focus on can make or break our research. It's not just about picking these outcomes, but also about anticipating all plausible results and being prepared for them. But it doesn't stop there. Measuring these outcomes is equally crucial. Who is measuring? When are they measuring? These are questions we grapple with, knowing that the answers can influence the success of our research. Then, we face the bias conundrum. Bias can creep in subtly, and if left unchecked, it has the potential to skew our results. That's why we devise strategies to prevent, minimize, or control for these biases. In the intricate maze of drug discovery, every chosen outcome, every measurement, and every control for bias plays a crucial role in finding the next breakthrough.